Okay, so welcome everyone to the first of a long series of planned lectures and events to celebrate the 75th year of India's independence. This year will also be the 60th year of the founding of the institute, so a few more events planned for that. The very first lecture today is as part of the popular series of lectures for the general public. So let me hand it over to my colleague Rahul to introduce today's speaker and moderate the session. Rahul.
Thanks, Vishwanath, and welcome everybody to our first event in the Azadi Ka Amrit Mahotsav series. Our speaker today is Professor Krishnaswamy, who many of you in the audience would know well. He retired from the School of Biotechnology, Madurai Kamaraj University a few years ago. He basically established and headed the Center for Excellence in Bioinformatics at NKU, which over many years under his leadership became one of the prominent bioinformatics centers in India. And he has himself worked on a variety of structural biological problems. And his students are basically prominent alumni all over India and the world. In addition, he has a strong interest in science outreach. And he was the previous president of the Tamil Nadu Science Forum, I think more than once, and is currently treasurer of the All India People's Science Network. For the past few years, he has been a visiting professor at the computational biology group at IMSc, where he has been teaching part of the protein course and generally keeping the atmosphere lively. So I'll hand it over to him now, Professor SK.
Yeah, thanks, Rahul, Vishwanath, Arijit, for this opportunity. And I think it's a good thing that we can have this talk. And I'll just share my screen, so I will go to the slides. So as you can see, my email IDs are given here. The one is a Gmail ID and the other one is a IMSc email ID. So just in case anybody wants to send in an email for questions or anything, feel free to do so. So this talk is related to the 2021 Nobel Prize in Physiology of Medicine that was awarded jointly to David Julius and Ardem Patapoutian. And before Ardem and David Julius and Ardem started their travel down this road, they were awarded just in September of last year, 2019. David Julius got this $3 million breakthrough prize for his work on the pain sensation. Subsequently, around October, the Rosenstiel Award from Brandeis University was given to both of them for this understanding of the touch, temperature, and pain. And then came the Kavli Nanoscience Prize on Neuroscience to discovery of the receptors for temperature and pressure. So they were sort of slated for the 2021 Nobel in a way. And you can see, therefore, that this discovery for receptors for temperature and touch, which actually forms one of the basic essential senses. We have our eyes and nose through which we can smell things, the eyes through which we can see, and the eyes are through photons, are converted to electrical signals in the nerves, and the nose, again, there are chemical receptors that catch and convert them to open up ion channels to get signals into the brain. Then we have the taste. Again, the taste buds receptors. These are also involving ion channels in a way that they convert the taste receptors, convert the chemical signal into the electrical signal, which can be conveyed to the brain. The hearing is again partly due to this work that we will see, the pressure sensors. But the most obvious thing that is there all for us is our sense of touch, the feeling that when you walk, you feel the ground, or when you reach out for a hot cup of coffee. In fact, that was the other title I had given. That obviously that is because of the fact that you're not only able to sense the temperature when you touch the mug, but the fact of reaching out is you're having a sense of perception of where your hand is, even without being obvious about it. And that arises actually from this the pressure sensors that were discovered by Ardem and his group. The fact that I can close my eyes and touch my nose, and the straight or around, I can do that. That's because of the fact that my sensors tell me the muscle stretches. It's sort of the pressure sensors trigger ion channels which give action potential down the nerve, which tells where my hand is. So it's something so innate, to all thing, and that is the reason why. And we, as you will see later, this pressure sensors seem to have a role to play in many other parts of internal organs also. And the other thing I would just like to point out is the asymmetry that we have been more and more noticing. That the Nobel in medicine was given 112 times to 224 individuals, and you can see the women form only 12 of them. So it's obviously there is something that is lacking there. In the sense that we need to give. It is not just a random thing. It's not just increasing the number of women in science, but making going forward and making sure that there are a lot of women who do deserve work, done work, which obviously deserves Nobel. But this year's Nobel for for touch and the temperature pain is really a recognition of the basic sciences. The fact that basic sciences, both David and Ardem are proponents of the fact that basic science is very essential for understanding and further research. So they are listening, and both of them did the groundbreaking work in the lab just around when they were mid-40s. David is about 12 years or so older than Ardem. But in 97, in David's lab, they got this heat-sensitive ion channels, and in 2010, in Scripps research in Ardem's lab, they got this Piezo receptor, identified that receptor for this. So the other interesting fact is that Ardem Patapoutian is born in Lebanon, and his mother tongue is Armenian, and the first sort of person of Armenian origin who has got this Nobel award. And if you look at just before I go into the topic, just that the Nobel is also asymmetric, not only in terms of gender, but also in terms of the location. You can see that these are all the Nobels, not only in medicine, but you can see the far outstrips US, far outstrips others. And this sort of begs a question why, of course, and the other interesting thing that has come up, as I said, Ardem is an immigrant who came from Lebanon when the war was there. He came at the age of 18. Apparently, a traumatic event of militants catching hold of him and then releasing him, that triggered his thing, and he said enough is enough, and he came over to the US. And so you can see the immigrants contributing quite a bit to almost more than 30% in the sciences to the Nobel category. And as one of the things could be that, as Ardem also says in his interviews, that oops, I went to the last slide. As Ardem says in the interviews, that it is because of the fact that he is very much, being an immigrant, he works hard and he doesn't take things for granted. And that's one of the things he says also during when he got the award. It turns out that apparently when he came to, here, he didn't really plan to be a scientist. He worked in English as an editor for a magazine, and he even wrote astrology pieces. And of course, he was doing a pizza delivery and so on. Then he joined for his life science thing, and he actually wanted to go into medical, laboratory type of thing. And it just so happened he joined the lab, and then he became interested in basic science, and then he continued in this path. So one important thing that I think, in my view, that this prize shows is that the importance of molecules in our the fact that we are all related, all organisms all over the earth. 150 years ago, Darwin showed that in terms of macroorganisms, but now we know at the level of molecules, this evolutionary relatedness, and this sense of sensing of the environment around us is done through molecules. The molecules that come from the environment and come impinge on our the organism, and then the organism responds by the molecules which are on the surface of the cells. And it is the molecules on the surface of the cells, the receptors, that actually trigger downward processes which make the organism realize whether it's hot or cold, or whether it is coming against a wall, or whatever the nature of the environment. So this is something I think which needs to be stressed, because in these times this relatedness is something very important. And as Feynman actually said, we are but a bag of molecules, and all organisms are but bags of molecules. So I will just, before I go into the thing, I'll just introduce a bit about the proteins. Since the general audience there, we have three types of proteins which are made up of amino acids. The proteins are encoded by genes, and you have the soluble globular protein like hemoglobin, which is involved in the carrying oxygen through the iron. And then a membrane protein such as porin, which sits across the membrane. It is this membrane protein, porin, is one of the things that my group worked on in Madurai Kamaraj University. In fact, we are the first group to determine a membrane protein structure in the country. And then there is this fibrous collagen protein like collagen, for which Gene Ramchandran is famous because he deduced the structure from his X-ray films, X-ray diffraction photos. So amongst these, the membrane is the one which sits on the membrane. The membrane protein is the one which sits on the membrane and is able to perceive outside molecules and then transmit what information further on. And if you look at the types of membrane proteins, the main one we focus on are the integral membrane proteins. Those which are having beta strands which cross the membrane, that's the porin. Others which we worked on. The other ones are the helical ones, which have alpha helices which span the membrane. Bacteriorhodopsin is a very important protein which is involved in the reception of sensing the photon and converting it into signals. So there are many membrane proteins, and you can see that the total structures in the protein database, if it's the order of 10 power 5, membrane proteins are the order of 10 power 3. And you can see more details on this. You can see that as of almost last week, number of protein structures is 160,000. Of which the one released in 2021, in spite of the pandemic, is about 8,400 something. But you can see that membrane protein structures, which are part of the PDB, but have been culled out separately by Stephen White and his group and put into a separate data set. Also, you can see that the unique ones are only 1,000 and odd, and the total ones about 5,000, much less than the ones that were released in 1 year itself of globular protein. So you see, our understanding of membrane proteins is far limited. And you can see the growth is fairly good, especially now with the advent of cryo-EM, which people like Natesh from ICER, who's here, who should be knowing that is very good in helping us to get more structures. It has also been recognized with the Nobel Committee. The 88 chemistry prize was given to Dysanov Hubel Michel for determining the photosynthetic reaction center, which is involved in sensing light. And then 97 to Boyer and Walker. Walker worked on SCOW. SCOW worked on the ATPase, which is really a huge protein that sits and converts thing. And then chemistry 2003. McKinnon came up with the ion channel structure, the potassium ion channel, which really tells how there is an asymmetry that can be established, because in a cell there is more of the sodium ions outside, and the inside is more of potassium ions and others. So you have a negative potential with respect to the outside. And what these ion channels do, as in the case of the ones which you're going to see, the temperature one or the pressure one, is they open up and allow a lot of cations to go inside. So the result of which is that the membrane depolarizes, and this once it goes above a certain level, it leads to an action potential, and it is that action potential which traverses down the neuron to further this thing. So the 2012 chemistry prize was given for GPCR, which are again membrane proteins. But then we know that this year the prize has been given for the Nobel in Physiology and Medicine, and this is not with chemistry. That's because apart from the fact that they did their groups, both David's group as well as Ardem's group, worked, collaborated with others and determined the structures of the heat receptor, the temperature dependent receptor, as well as the pressure dependent receptor. But they also have done a lot of other things relating to how the functionally these molecules and in characterizing the various ways of action of these molecules. So in nervous system also has received a lot of thing in the Nobel in Physiology of Medicine, starting from the early ones of Golgi and Kajal in 1906, where they really gave the structure of the nervous system, and then subsequently Adrian and Sherrington, who also told about the somatosensory neurons, which are the ones which really are involved in transferring the sensation of pain and pressure into inside. And then in 38, the Cornel Heymans came up with the discovering the vagus nerve reflex. I'm mentioning this in particular because this is one of the things which Ardem and his group have really shown how the temperature, the pressure receptor, piezo receptor is involved in this respiratory reflex that was described by Herring and Broer, which Heymans shown is involved using the vagus nerve. But and then subsequently, people both Erlanger and Gasser showed there is differentiation of the somatosensory thing. So there are a lot of different regions that they serve. And then came in '63 the famous work by Hodgkin, Huxley, and Nichols, where they came up with a way of describing this, how the action potential traverses down, came with a mathematical model. And what is interesting is at that time, even though there was nothing known about the ion channels, they came up with a proposition that there has to be these ion channels which are there, which will change the ionic balance inside the cell. And so it was really it is these ion channels that subsequently McKinnon and now David and Ardem are coming up discovering. So in this line we have now the 2021 prize related to this fact that the sensory neurons. This is just a from Ardem's review, which shows how, if you look at the nervous system, it's not just the brain. There are a lot of things with the spinal cord, and from the spinal cord you have these, for example, the dorsal root ganglion that goes, and it has ends up with various receptors for temperature, pain, and then there are this mechanoreceptors. The proprioceptors are the ones which sort of give you an idea about where your limbs are, even without having to look at them. So these receptors give the inputs, And then subsequently over a synapse it transferred to other thing which go on to the efferent or the motor neurons, which can make the action. If there's a fire, to take your hand away from the fire and so on. These are this is a cross section of the skin which shows epidermis and the dermis. You can see that the green ones are the neurons with the free nerve endings, which are part of your the temperature pain receptors, and the red ones are the ones which are part of the mechanoreceptors. Mechanoreceptors are all connected to bodies like the hair cell follicles and so on, because when the hair moves, you feel the pressure, the movement of the hair triggers sensation of the wind that goes across. And these free ending ones are the ones which are involved, the pain receptors which are there, the temperature receptors. That is why when your skin there is a itching on the skin, or something falls on your skin, some hot chili touches on the skin, there is a burning sensation that comes from this. This at the bottom is just a section showing staining, showing that the free nerve endings are actually on the epidermis. The green ones are the free nerve endings which are visualized in staining. So this is sort of also tells you that the nerve cell, unlike our normal cells you see in the textbook diagrams, is not just a ball, or a round thing with it is a sort of very asymmetric, elongated thing. And there is an asymmetry in the sense that the entire axon is more or less covered with myelinated thing to make sure that the action potential travels well. And the terminal parts of this is where the receptors come on that. So it's a very asymmetric cell, this nerve cell. And you can see that at the end, the terminal parts is where your receptors for the pain or for the mechanoreceptors are placed. And for example, this there again, one of Ardem's talk is that if you have a 6.5 feet person, like my son, for example, then he is likely to have a neuron which will be more than 5.5 feet long. One of the neurons, which is because it has to go all the way down to the toe and then also to the arm and so on. So these are some of the long ones, and there are nice pictures of how in a zebrafish the entire neurons take about 16 days to go and spread themselves over across different parts of the body. So the nerve endings of the neurons are where the action is taking place. So look at thermo. First, I'll deal with the thermo and chemosensitive ones, ion channels by David, Julius, and group, and then move on to the pressure-sensitive receptors by Ardem and. So if you look at the chemosensitive channels, ion channels, the fact that there is something involved was known earlier. You can see, there is a way back in the 50s, the gustatory sweating is the sweating on your head, forehead, and so on when you eat something hot, or something noxious thing falls on it. So that sweating is what was described, that it is something to do with the nerve, as what was earlier itself thought. And then specifically the capsaicin, which is a component in chili, that acts on sensory nerves was shown, and then that it induces ionic currents, shown in the 90s, and then about 97, the same time when Ardem's group characterizes capsaicin dependent receptor. The group, the Reckling and Levin, showed that noxious heat, that is like at around 40, slightly warm or little more than warm, when it can get scalded, that also activates ion channels. Okay, so now David Julius, it turns out he seems to be a fairly simple man, and he is very motivated, and of course you can see his tweet about how he's acknowledging his teachers and so on. And his Holy Ingram Ham is his wife. She's also a scientist, and so one of the stories that David Julius has told when he got the breakthrough award in 2018-19 was that apparently he got this idea of the fact that this noxious heat, which was shown by Levin and others, could be related to the capsaicin or chili sensation. And when he walked in a supermarket and saw this whole aisle full of chili, various stuff that deal with chili, and so he said that he must look into it, and apparently Holy Ingram also said yes, you should go ahead and so on. That's how he started the idea that I will skip this slide for now because it's a very detailed slide. Basically, I put the slide to tell that Katarina, who is the postdoc who was involved in this work. He is now faculty in Johns Hopkins University, but he is the one who really came up with, identified the receptor that caused the thing. I'll leave this for the questions. If there are any particular questions, I'll come into the slide. But there's a more simplified version of the slide put out by the Nobel press release. Is that what basically Katarina and Ardem, David Julius did was that they looked at this. They looked at various DNA fragments which are likely to be involved in this having this capsaicin or related genes, receptor genes. And so they put the cDNA into the cell to see whether this is a cell which normally doesn't have any response to capsaicin. So they kept putting various DNA fragments into that to see whether which fragment makes it respond to capsaicin. Capsaicin is this molecule which comes from chili. So that once they identified that, they also subsequently showed that this receptor, which they narrowed down on this receptor called TRPV1, transient response potential V1. V is for vanilloid type thing. It was earlier called VR1. They changed this name to this. So this is also causes noxious heat. That's one of the things earlier in the slide, which showed that he was able to show that if you put this thing into a cell and then at around 40, say you can do a patch clamp to look at the currents that are induced, and you can see the currents being induced when you have just above 40 degrees centigrade. So that is the part which it's a laborious work, basically taking fragments from the dorsal root ganglion cell of the genome, making cDNA of that, various overlapping fragments, and then putting the cDNA fragments, and then whichever cDNA fragment responded, then they had to fine chop that cDNA fragment to make smaller fragments in order to finally come up with this thing. So that's what they did, and they also showed, as I said, that temperature also works. So basically, when they put capsaicin or something, the channel which closed initially opens up, and as I said, it's a fairly non-selective cationic channel. So you have a flood of cations coming inside, which depolarizes the membrane potential, makes it more positive, and then causes an action potential, and that's how the thing goes. So apart from this, they also characterized other receptors, TRPA1, which is involved with mustard seed, and TRPM3, and also TRPM8, which is involved with the cold sensation, menthol or mint, from what is there in mint leaves. So they showed that that is open to this. Okay, but this is where it gets interesting, that if you look at it now, there's a review which sort of tells all the thermoreceptors which are there. There's a new category of thermoreceptors which people have identified called K2Ps, which are having slightly different number of integral the helices which cross the membrane compared to these. So it's a smaller one than the TRP membrane proteins. And if you look at this, TRPV1 was one which was characterized by David Julius group in '97, and that is the basis for the Nobel thing. Of course, he has done subsequently work along these lines. And you can see that in 2002, David Julius also characterized this TRPM8. There is a menthol thing, or the cold-related one, and finally clinched it in 2007, a back-to-back publication along with Ardem in 2007, who also characterized this thing as a receptor that is sensitive to both cold as well as the menthol. And they also characterize this TRPA1, which again is it opens up a variety of wintergreen and a lot of noxious substances. And this was in 2004. Both David's group and Ardem's group characterized at the same time. And along with this, a group by Voigt's Singh, they in 2011 characterized this other this thing called TRPM3. And for quite some time, people couldn't figure out in the rodents and mice, for example, if you take knockouts which don't have TRPV1, they were still feeling the heat and capsaicin. Or you take out TRPA1, again they were still feeling this thing. So then Voigt's group showed that if you take all three out, TRPM3, TRPV1 and TRPA1, then the mice don't become sensitive to the heat or the capsaicin type of thing. So that now it's known that there are many things in different organisms, the proportion differs. So it's basically not a single thing that allows you to sense all these noxious heat of the substances. And we just go on to look at the structure of these channels. That one of the channels called there are TRPV1 to TRPV6 is there. All six have been characterized with the first one. TRPV1 was characterized by Katarina with David Julius and his collaborators. But now all six have been done, and with various compounds, capsaicin. There's also a spider venom that sort of that's the reason when a spider bites, you feel this fiery sensation, and that's because the spider venom is a toxin that goes and blocks this channel. It keeps the channel always in the open state, just as capsaicin, when it binds, or any of the other noxious substances, when they bind to the channel, they keep it in an open state all the time. So then this comes in, and these are not only sensitive to these noxious substances. It turns out they are sensitive to even anti-inflammatory compounds that are produced when your body has any inflammation. So some of these also trigger this thing. So that's the reason why when you have this inflammation and so on, that region becomes a little painful, and also this feeling of heat comes there. And that is because it goes and keeps this channel open. And this is a view cross section. This is the membrane part, the gray lines, supposed membrane part, because it is deduced, and the outside and the inside. And they found there are two gates. There are two gates which are present. The potassium channel by McKinnon had a single gate. Here there are two gates involved, and they seem to be fairly non-specific in the sense that they allow a variety of cations to come through that. And there's a view 90 degrees of that. So it's a view looking down, as you would see the ion would see looking when it goes into the pore, when it goes into the through the membrane. Okay, so now this what is interesting is that in spite of all this work, this probably a good thing for computational biology people do, in that it's still not known how heat actually triggers this thing open. There is no real thing done on showing. It's been shown experimentally that heat opens this channel, right? But because heat being temperature being what it is, you can't trap it in a particular state at that. So there's no way to keep the channel open in a trapped state at a particular temperature at say 43 degrees. Obviously, it opens up because it's very similar to what happens with capsaicin, and capsaicin opens up this channel, binds to some region here and keeps it open. So this is still not known. So what is the effect of temperature? How does a temperature really open up this channel is something that people still have no idea about. They know that it does, but they don't know how. Okay, so now I will close with that thing. That is one set of channels, the TRP channels, and now also the TREK TREK channels, which are slightly smaller. But basically, these channels sense some noxious, some chemical substances, and they also respond to either heat or cold. They open up during cold or heat, and allow the ions to come in, the action potential to be generated. And because there is a relationship between the pain and the thing, that's why this pain sensitive channels, they are also involved in that. So one line of work that David Julius and his group are now following is to try and see whether this idea can be used to come up with non-opiate painkillers. No, now we have only painkillers which either relieve by morphine related or analgesics from aspirin and so on. So one of the things that David Julius has said during his interview is also that he tries to look into nature and see what sort of compounds can be used to try and activate various processes, and that's how we started on this. And he hopes to follow it up with coming up with using these things to see whether there can be some way of blocking such things, so that preventing. There is one type of pain called Allodina, where it's a very severe pain, and that's what people want to try and protect. Okay, now coming to the mechanosensitive channels. The mechanosensitive channels again were known for quite some time. Way back in the 70s, there was a nice work which showed that the hair in the bullfrogs, they are the ones that they're responsible for this mechanical simulation of the hair. That is what is causing the membrane depolarization. They didn't know how, but they were able to show that. And then mechanosensitive channels existed in E. coli. People have been able to characterize that what is called MSC-S or MSC-L, these genes that are there, and that's the reason why E. coli also feels the pressure, turbid pressure. If you obviously change the osmotic thing, it becomes there has to be a way of preventing it from bursting. So that is what happens to that. And then 99, the group showed that these mechanosensitive ion currents are present also in rat dorsal root ganglion neurons, and that's what triggered with Ardem's work, where what they did is they screen is frozen. Yeah, okay. So now this is the one by Ardem. Essentially, this discriminative touch, very fine sense of discrimination thing, and then what is called affective touch, like hugging and so on, or proprioception, the fact that you can play violin or flute or something without having to really see where your hand is going. So that all this is done because of these pressure receptors. And there's a nice talk with Ardem has given in 2018 in Scripps Research, which the title is "The Force is with You." And of course, this on the left here is from his Twitter thing, where he tweeted the photograph of his after when he got the news of the Nobel. That's his son Lucas, 14 years, who's there with him. There's this by now fairly famous, or everybody would have heard the story, that he had his phone on do not disturb mode, and therefore when the Nobel committee called, he didn't go through. Then they called his 92-year-old father, who was in Los Angeles, and fairly father got bugged. They wouldn't tell him why they were calling. So then his father finally called, because in do not disturb apparently favorites can call. So he called Ardem, and then finally he got to see the Nobel Prize announcement sitting on the bed with his son. So that's the story there. And so what happens is that Ardem's lab is also good in some diversity. I just want to He's a very nice guy to see, overall see, and the his approach of bringing in diversity is also good. You can see people from all over the place pointed by the country of birth, and you can see one mark in green as women and red as men. And so you can see here that this one woman from India, it turns out to be from Bangalore, and she went and did a PhD in Buffalo, and then went did a postdoc with Ardem, and she is one of the people who characterized from the capsaicin-induced feelings in mice. She is now a faculty in Portland, Oregon, working on interesting stuff. For example, there are flycatcher plants. You would have seen these plants, fly-catching plants, the Venus flytrap. How are their hairs sensitive, and how do they detect? It turns out to be due to these piezo receptors, and so that's what she's working on now to try and get more details on that. So they've actually characterized one of these receptors in the Venus flytrap plant. And this slide again I will skip. It's a heavy slide which just tells the reason I put it is again for questions. So I just want to point out here, Bertrand Coste is a postdoc who did the work, took 1.5 years to find this thing, and then he's a faculty in Marseille, France. And what he did was actually identify these neuro-2A cells, which this is easier shown here in the slide from the Nobel press release. You can see what they essentially did is that they took these neuro-2A cells which didn't have the mechanoreception, which had this mechanoreception, and then what they did was they silenced various genes which are in that using siRNA method. So they silenced one gene after another, and they had 72 candidates, and 71 candidates gave nothing. So you put a mechanical force in the cell, you measure using patch clamp the current that is generated. So 71 candidates, nothing. They all, you silence them, they still show mechanical force response. The cell shows mechanical force response. So that took Bertrand Coste a year and almost half, right? And the 72nd, the last candidate, was the one which, when they silenced, they found that if you push the cell, there's no current that's generated. So, so this is, as he says in one of the presentations, Ardem says, it takes me just 30 seconds or so to tell this, but it took my postdoc 1.5 years of no results to get up to this. And what is interesting is also now the 72nd thing has become such a catch thing, and all these things about the Nobel thing. There's a description of how 71 didn't work and so on. But when you actually go to the paper, the paper is so dry. It maybe it says how we must really change the way we write our articles, so that there can be a little more life in them, which people can relate to. So this is what happens in the Piezo. What he characterized first was this Piezo 2, and the Piezo 2 is and subsequently the Piezo 1. Both of them are channels which, when you press, they're closed initially, but when you give a mechanical force in the membrane, it opens up and then creates this inflow of non-specific cations, mostly calcium and sodium, and so because of this there's an action potential, and then you are able to see feel the touch, the hug, and this because they are linked to our muscles. When I stretch my arm, the stretching of the muscle causes the action potential, which then tells, okay, this the arm is here or arm is going, and so on. So all that, the fact, when we walk, we take it effortlessly, but that walking, subsequently, Ardem and his group, in collaboration with the NIH group, has shown that there are people who are Piezo 2 deficient, and these people apparently cannot walk properly, because they don't have, unless they get inputs from other senses like the visual sense, they really are not able to, you blindfold them and think they just can't walk properly. And that a lot of times has been misdiagnosed by clinicians as motor neuron disease or various other things. But now they see that when they screen the genomes for this Piezo 2, they find that they are Piezo 2 deficient. So it seems to be that's one, right off. This tells you things which we didn't earlier even think of. There are two piezo proteins which have been characterized in human. They are slightly different, and you can see that they are present in a variety of organisms. And subsequently, piezos have been characterized in plants, as I said, flytrap, and even in some protozoa and so on, pathogenic protozoa and so on. It is there. So this is a cryo-EM structure of Piezo 1. Piezo 2 structure has not been done yet. You can see that this is very different from normal membrane protein. These are the three arms which are there, like a trimer, which is there. So this trimer-like thing has like a cup. There is a domain on top, which you can see when it's rotating on the top here. So that's called the CED domain, this part here. And what is shown in the cartoon sketch on the left side is how normally this is how it's thought to sit in the membrane. We don't know how actually these helices should be in the membrane. They are transmembrane helices. So that they are sitting like this. And then when there is a stretch or a shear, when the membrane stretches for whatever reason, because there are actin filaments attached to them or because of something else, then they pull this and they open this up, and then you have these ions that go inside, right? So this again, this part is, there are two models how the stretching can happen. Both the two, which of these models actually is not yet been established, right? And again, this is something very interesting. And a lot of people working in virtual reality apparently are also talking to this Ardem's group about how whether some sensors can be made using these things in order for people to get more realistic feelings of touch. And if you look at this, mechanical allodina is the one which causes neuropathic pain, sunburn, variety of sensations which are there, which is there. So this is what Shweta Murthy, I told you earlier, she worked on in that was a postdoc. And what she was able to show is that if you look at capsaicin, capsaicin induces allodina in mice, which is mediated by Piezo 2, not the TRPA1. No. You keep this in mind. See, it's not the thing. So what happens is Piezo 1 seems to be along neurons, which are on the surface, the skin, and those neurons which come to that. But Piezo 2 seems to be present in a variety of internal organs. It's there in the heart, the bladder, even to do things. So it is present in a variety of places inside the organism. Also different organs. And what Murthy showed is that if you take mice which have this Piezo 2 knockout, the wild type, and then the knockout mice where the Piezo 2 doesn't exist, it's not functional. There, if you apply capsaicin on the mice, the feet, then they don't respond. Whereas if you apply to other mice which have working Piezo 2, they respond immediately. There is a response on that. So whereas here in Piezo 2 deficient mice, the response is not there. Of course, they did a variety of other things also show that what happens in nerve injury and so on, other cases. Okay, now this is the one I said that they also have shown in Piezo 2 deficient humans work with NIH thing, where one nice thing which they also show is that it's called tactile allodina, where you take an arm and then you put add capsaicin, and then after that you touch. You decide whether the capsaicin was put on the A part or the B part, right? And for a control, they have a laser Doppler imaging, which tells where the actual capsaicin was put. And if they look at those people who do not have this Piezo2 or who are deficient in Piezo2, that gene is not there or is not in enough copies, then they find that there's a loss of function which is there, and such people are not able to tell where actually it was applied. And if you ask them to choose, they choose randomly, and that's why the touch test gives you about 50%, which is random. Whereas if for those people who are not deficient, they are always able to get it right where the thing was put. And these people also, when you ask them to walk, they just walk completely randomly. They're not able to walk in a straight line, and it was really surprising to them that it's all because of this Piezo2 not being there in the gene. So now, if you look at the mechano sensation thing, what they call is interoception, that is sensing the internal organs. This is perioception is the one where you sense the external world and how the external organs, the arms and limbs and head and so on. The internal organs also you can sing with this mechano sensation and somato thing. So you have the touch sensation, pain, respiration, baroreflex, bladder, stomach. These are some of places where people have shown from both Ardem's group and others that these Piezo2 receptors are there, and that is what causes this. When you have respiration, as I said earlier, they had this thing of when you breathe, your lungs inflate, but they cannot. If they inflate, they'll burst. What prevents them from bursting? Because there is a pressure to put them back, and that is because of this pressure receptors. The baroreflex has just shown same thing they have shown with bladder also. Your bladder becomes full, and that sensation is because of the Piezo2 pressure receptor that tells you, okay, your bladder is full. And same thing for the stomach. When you eat too much and you're feeling a full stomach, that's because of the Piezo2 receptors. So the baroreflex is something interesting also. I just show a little more about that. It says they maintain blood pressure. The fact that when you stand up, when I'm sitting and I suddenly stand up, then there is a drop in my thing. So this, if there is an elevation of the blood pressure that is there in the legs or so on, when I sing, that conveys to the vascular wall. And in the vascular wall, this triggers the piezo receptors. So that causes the action potential, which then traverses down and tells your brain, okay, now seeing the control of the heart is changed, and then the heart rate goes down and your BP goes down. Right. So this is something. So if you have failure of the baroreceptor function due to the piezo2 receptors either not working or having then you can have arrhythmias, you can have hypertension and a variety of things. And this is an interesting thing. I found that there is a guy called Eric Topol whose tweet I've given here, who's also from Scripps Research, but he goes around taking any publication. For example, this obviously got published in Science by Ardem's group. I think this is a leader, a review of that which was put by MK. But the original article also came in Science. And so he tweets this saying that, this is more than 1852, from then on it's not known, a 170-year-old problem which has been solved by the group in Scripps. So I think having such, he does this only, and this is a good outreach thing which can be done, rather than expect scientists to go around doing their own thing, having people like this who can tweet out very significant findings, and when they are published and so on, makes good sense. And this is something which the baroreflex. This was shown for piezo deficient mice. Interestingly, in this baroreflex, unlike in the other cases, both piezo 1 and piezo 2 have to be knocked out. If you knock out only piezo 1 or piezo 2, because obviously, I guess this blood pressure is such an important thing that there's a sort of standby thing. So if you knock out both piezo 1 and piezo 2, then you find that on the right you can see the baroreflex goes down on that. So piezo deficient mice where both 1 and 2 are not there, they have high blood pressure variability and they are hypertensive. So this is something again which they would like to use to see whether people with heart conditions can be relooked on this. Okay, so now sort of coming to the end, that you can see that in sensory neurons you have this pain, baroreflex failure, inner ear. This can cause deafness. Piezo deficiency can cause deafness, and in blood vessels, the piezo receptors are responsible for keeping the tension, and so lack of them can cause various diseases. Again, bone formation, osteoporosis is also linked to this, and I'll touch a little more about the red blood cells, where again in red blood cells also these receptors are there, and they are sort of seem to be involved in sickle cell and have some sort of a functional thing in malaria protection, and lung, eye, kidney again, and then mutations in Piezo1. They can cause such genetic diseases as lymphatic dysplasia, where you have the swelling up of the legs, which is there. So this has been shown. This was shown by a different group. But there's a whole list of medical conditions subsequently people have shown that where this absence of Piezo1 or Piezo2 seem to be causing or mutations in them are causing diseases. Okay, so for this red blood cell thing, normal blood cells are shown here on the top part here. There's something called hereditary xerocytosis, which where the blood cells, some of the blood cells take up these funny different shapes. They are dehydrated, so they lose their shape a bit. So there's dehydration is there. This is a very rare case. So about only 100 cases have been reported. And all of them have different gain of function mutations in Piezo1. So earlier what all we've been seeing are loss of function, where Piezo1 is deleted or mutated on this. Here there is a gain of function. So Piezo1 has a mutation which makes it act more. That is, it sort of increases the amount of ions that are brought in, or the channel is kept longer open and so on. So this, they've shown that if you have genetic conditions associated with malarial resistance, they also affect the red blood cell volume, right? And this Ardem's group of postdoc has shown that he sampled various African population, both in Africa as well as Afro-Americans, and showed that about 1/3 of them have this gain of function mutation. Sorry for the spelling mistake. It's prevalent in the African population, which is there. So this is the almost last slide on that, which just shows that that if you take the wild type, normal red blood cells are there, and you introduce Plasmodium falciparum, which causes malaria, mice get cerebral malaria. And then you make a Piezo1 gain of function mutant and put it into this mice. Then they have dehydrated RBCs. Not all of them are dehydrated. No, some of them are this thing. And if you expose them to Plasmodium falciparum, they seem to be protected from malaria. And same thing has been shown in humans also, of course, not with experiments like this, but in real thing, by sampling the genome of the population. You see, in wild type, you get normal blood cells, and then you expose them to falciparum, you get a high infection rate. But as I said, about 1/3 of African and Americans with Afro-American origin, African origin, they have this particular thing. In this protein, the 756th amino acid, glutamic acid, is deleted. No, there is a thing that's a deletion mutant, and that gives it a gain of function. So this Piezo1 acts more than, or increases the amount of current flow and so on. So, but this also leads to dehydrated RBC, because RBCs also have this Piezo1, and that's a, so with this dehydrated one, these people who have such a mutant in their genomes, when they are exposed to Plasmodium falciparum, they seem to have low infection rates. So this is something that very recently, and that's probably a reason why the Nobel Committee chose this thing for us, because a lot of large scale applications and so on, which people are visibly seeing because of the Piezo1. And so this is the last slide, which is just telling you that you have temperature and heat pain all coming from a family of receptors, the original of which was deduced by David Julius and his group, and they are to do with maintaining the body temperature, inflammatory pain, protective reflexes, so on. And then the touch, proprioception, and internal sensing. That because of the Piezo receptors, which was characterized by Ardem and his group, where you can see they're related to mechanical pain, urination, respiration, blood pressure, skeletal remodeling. There's a whole lot of things that are involved in medicine here, which people both in new characterization of diseases, which people earlier thought was due to something else. Now they are realizing is because of these absence or changes in these receptors. And then whether you can come up with medication which can help in some manner, maybe a topical application which will relieve the pain felt in a certain area and so on. So that's it with me. Thank you for this thing. So this is my last slide and I'm done.
Okay, thank you, SK, for the very interesting talk. We'll take questions now. We have already received a few questions on chat and on YouTube. I just want to mention questions in Tamil are also allowed. We'll start with the first few that we received. There's a comment from somebody, not a question, that the skew towards immigrants could be because of the high immigrant population in the US, which is the country with the largest number of winners. And then there are two related questions. Is there a different decay rate for the different stimulants such that ion channels close at different rates? And is the concentration dependent, that's kinetic, or is it only related to structure of the stimulant molecule?
Yeah, there are two parts to the question. No, one is the rates of decay are fairly the same. There are not much difference in the different channels. And it is a structure of the molecule that sort of the noxious molecule that activates. So it is of course there is a the number of channels that need to be kept open will depend on the concentration of your noxious substance or the heat. But other than that, because only if sufficient number of channels are open, then your change in depolarization will cause something above the will trigger an action potential when it goes above the threshold potential. So for that there has to be enough concentration. Otherwise, as far as I could make out, there's nothing in the kinetics which makes a difference.
Okay.
So I'm not able to see all the questions, Rahul. You are reading them out. No, I have got one question on WhatsApp from Varuni on Tamil.
No, there are quite a few questions on WhatsApp, but I will read them out to you.
Okay, there's one on Tamil which is there. Should I read?
Yes, if you want to go ahead with that one, you can go ahead with that.
Yeah, I got only one which Varuni has sent. It's called something from Arun.
Yes, go ahead.
It is கீற்றை உணர முடியாது, கீற்றை வைத்து வெப்பத்தை தடுக்கத்தான் முடியும். பெரிசாகிறதை காண முடியும், அழுத்தத்தை ஏற்படுத்தி பெரிசாகிறதை காணலாம். Well, it's really not a question, it's a comment.
Right.
Yeah, so
so okay, so more
that's
questions.
The only one in Tamil came here.
That's the only one in Tamil so far. And there are several others which should be there on your WhatsApp, but I'll read them out. One is, what is the typical distance between pain sensors, their surface density, say? This is from Ghanshyam Datte.
Okay, since you're reading out, I'm not trying to look into the WhatsApp. I'm not able to.
Okay, yeah, I'll read that out.
Okay, thanks. Yeah. Sorry, I didn't get that question which you said.
What is the typical distance between pain sensors, their surface density, for example?
I don't know, and I don't think that is known in the sense that there's nowhere they talk of what is the number of Piezo 1 receptors which are there on the ends of the nerves, or for example on the walls of the artery and so on. So I don't know. and I have not come across any such information. There must be some number, but I don't know. Yeah.
Thanks. Next question is from Bhaskaran, our colleague. Do heat shock proteins play any role in heat sensing?
No, really. Heat shock proteins play a role in sort of helping other proteins to either fold or to remove other proteins which are thing. So they are sort of not the cause of thing. The result of some shock to the system.
Okay, thanks. And the last question that we have here, please feel free to type in more questions on YouTube or on the Zoom. This question is, what is used to apply mechanical force on a cell? How is the force magnitude controlled?
It's actually it's like a pipette tip that is used to poke the cell. So I think it's just manually done. From whatever I could make out, I don't think there is a specific device that tells about the force. It's not like a single molecule type of thing. And that. So as far as I know, it's just a simple mechanical perturbation given on that. There must be some, I think that is not characterized. What is the amount of force required or pressure required for the receptors to move and so on, to open up? I think that is not, there's no characterization of that quantity. Yeah.
Okay, I think that's all. There are no more questions, it seems. So let's all thank Professor Krishnaswamy. Okay, sorry, there is one more question, two more questions that have just come in.
I think in slide 17, somebody has put
Yes, so the one before that, there was one slide which mentioned that asthma and deafness are connected to the lack of Piezo 2 or Piezo 1. Could you please elaborate on that? This is from Atul Krishna.
Yeah. So it's that is one of the things that there's not much work that is there done on the deafness and the asthma, but one of the things that they are looking into is that what would be the effect of the absence of these on the audit. Obviously, hearing process is a mechanical stimulation. So that would have to be triggered by the piezo receptors. And the asthma, again, because it is related to the lung expansion and the physical movement of the lung, which again is controlled by the piezo receptors. But there is not any work that, like in the case of baroreceptor or the urination, there is no work that is published on this asthma and deafness. It's just that they are looking into it. Yeah.
Okay, next question. This is from L. Ramya. Thank you so for an interesting talk. In slide 17, you mentioned that when temperature increases, channel opens. Does this membrane curvature lead to the conformational change in the protein?
What is 17? I just can I just look at my slide?
Yeah, you can go ahead.
This one, I'm still sharing the slide, so yeah.
Yes, we can see your slide.
Yeah, but this is my 17 and that 17 doesn't match. Yeah, I think.
No, this is numbered 17 at the bottom. It must be what she means. So what you're saying is when temperature increases, the channel opens. And does membrane curvature lead to a conformational change in protein?
Okay, so here the membrane curvature, that's the other one which I put. This is the TRPV channel.
Okay.
Here in the TRPV channel, there is no, the membrane curvature is not really coming into the picture. It's just that because of the heat, the channel opens. And as I said earlier, they don't have a structure where it is trapped at different temperature state or something. They have, because of reasons of experimental thing, they have only different states which are captured by chemical, chemicals which bind and trap it in a particular state. In those, there is increase in current, but whether temperature, I guess the question is the temperature would cause membrane changes, and whether that is involved in, I don't know, maybe that's related to the piezo one. No, the piezo temperature. No, because as we saw, noxious heat or the pain is not only related to the TRP channels, they're also related to the piezo channels. So maybe that's where the thing comes in that. No, but for these are something which obviously right now a computational thing can be done. Experimentally, we are far away from making such measurements, I think.
Okay, I think so. There are two more questions. We'll take these and perhaps stop. One is from Anandi. Are people who are tolerant to hot food, meaning spicy, karam, also tolerant to hot water?
I don't know really. I mean, this is, I guess, it is two things. One is that the taste is, I mean, they're not here looking into the taste aspect, right? That is not the receptors and the taste buds. It's when you put chili capsaicin on the arm, or on the face or something like that, or on the lips, right? It's not the taste of the hotness and the hot water. So I don't know. I mean, it's probably a good project. You should work with Shweta Murthy, I think, to get it done. Yeah.
There's a related comment from Ramakrishnan on YouTube. Tongue feels heat and the tastes of chilies as similar, but as you said, taste receptors are different.
Yeah.
Okay. Last question from Natesh. Is there an explanation for how pressure on cell surface at an adjacent point to piezo is transferred to the piezo channel?
No, no. As you said, it's there are just two models. One model which says that it is flat. I mean, I showed you the second model. There's one model which says the thing is flat, and once you put a pressure, then there is a change on that. The other model is that it is curved, and then you put a pressure and it flattens and opens up this way. So at what distance away from the channel, away from the receptor, one has to put the pressure, that I not there anywhere, and maybe it's not characterized, because it needs people really need to work with a small number of molecules in a small patch to do this thing. So yeah, I guess these are questions. There's a lot of questions obviously come up, and from 2010 these channels have been characterized, and the one mechanosensitive channels in equali have been characterized before that, but even for that we really do not know. No, obviously E. coli also feels the heat. No, E. coli also feels the pressure. So even there we do not know the answers to the questions that are asked now. So yeah. But one thing which is there is that what the Nobel has done is put new or made a bigger solar sodium lamp on the whole thing, so that people, there are more questions that people ask in this area, much neglected area, which obviously is involving all organisms, and the fact that we are able to survive the world around us is related to that.
Okay, thanks very much, SK, for this wonderful talk. Vishwanath, do you want to say a couple of words before we wind up?
Nothing much, except thanks for kicking off this series of lectures.
My pleasure. Yeah, it's nice.
Yeah, so it was nice to have this lecture on, I mean, the topical thing of the Nobel Prize on something so interesting as this.
And I guess everybody has a touch for it because of the fact that you touched.
Okay, thanks, SK.
Thanks. And one last thing that this is, as Vishnu said, the first in the series, we will have another talk on the physics Nobels shortly. Keep a watch on our Twitter feed or Facebook feed and so on. We'll be having, in fact, a couple of days from now, we have a talk by Professor Ardem. I think it's on October 21st. So there'll be many such events coming up, and keep an eye on our social media. Thanks, everybody.